NCRII will be presenting you the working and the whole procedure of ETC process in detail. You will forget anything but one statement, that mitochondria is the powerhouse of the cell and that cannot be true without ETC process. Now, what is ETC? Aerobic cellular respiration, as we all aware of, has three parts. Glycolysis, where glucose basically get metabolized into two molecules of pyruvate. This pyruvate oxidizes to acetylcholine enzyme A and gives molecule like CO2 and NADH. Then these molecules go through Krebs cycle, and through this process NADH, FADH2 and CO2 and ATP are produced. Now these molecules will go to the next step, and that is oxidation phosphorylation, which has two parts, ETC and chemosis. ETC basically consists of series of 5 protein complexes along with other constituent couple redox reactions. So today we all are going to perform ETC procedure in front of you in detail. So my batchmates Sejal, Tanu, Bhumi, Tripti, Vansh Gunavat, Varisha, Yash, Umang and Vansh Mangawa, they all are part of cell membrane, inner and outer. Vrinda is going to be performing ubiquinone part. Now I would like to present the anchors of the complex 1 NADH dehydrogenase, Abhishek and advanced stage to carry on the presentation.
Good afternoon to all. We Group 1 are here to present Complex 1 in front of you. As we know, Complex 1 is also known as NADH ubiquinone oxidoreductase. It is located in the inner mitochondrial membrane. It is most electronegative with the redox potential of minus 0.32 volt. It comprises over 42 proteins and FMN, that is flavin mononucleotide, which is a derivative of vitamin B2. It also contains cluster of iron sulfur center in which ferrous ion is attached either with sulfur atom of cysteine residue of protein or with inorganic sulfur atom or both.
So now my batchmates are going to demonstrate how NADH ubiquinone oxidoreductase works through a role play. As the NADH readily available in the mitochondrial matrix, it will transfer its electron to FMN and itself get oxidized to NAD. The electron will further pass from FMN to 8FeS. And then to ubiquinone. And simultaneously, protons are pumped from matrix to intermembrane space. So that's all about NADH ubiquinone oxidoreductase. Now group 2 will come to demonstrate the further process.
Good afternoon to one and all. Today I, Diya,
and I, Durgesh,
are here to demonstrate complex 2 of ETC. Complex 2, as we all know, is also known as succinate dehydrogenase, is a part of ETC. In ETC, complex 2 transfers electron from succinate to ubiquinone. This process contributes to the generation of a proton gradient, driving ATP synthesis during oxidative phosphorylation. Complex 2 is unique in the ETC as it directly participates in the Krebs cycle, highlighting the intimate connection between cellular respiration process. Succinate dehydrogenase is a key enzyme in both the Krebs cycle and the ETC in cellular respiration.
location. It is embedded in the inner mitochondrial It is located in the matrix of mitochondria. In Krebs cycle, complex 2 catalyzes the conversion of succinate to fumarate while transferring electrons to its cofactor flavin adenine dinucleotide, also known as FAD, forming FADH2. This is crucial step in the cycle. Connections to ETC. The electron carried by FADH2 from the Krebs cycle are then transferred to complex 2. From there, the electron passes through a series of iron sulfur clusters in the complex and eventually to ubiquinone, which becomes reduced to QH2. Thank you.
As the electron moves through the complex 2, there is no direct pumping of protons across the inner mitochondrial membrane as in complex 1 and 3. However, the reduction of ubiquinone contributes to the proton gradient by providing electron to the next complexes in the chain. Now hand over to complex 3.
Good afternoon, we are the complex 3, also known as.
Q cytochrome C oxidoreductase or cytochrome PC1 complex.
Now let's understand what the components of cytochrome C are.
First, cytochrome B and cytochrome C1, they are chemically proteins contains a heme group that is porphyrin ring with iron. This cytochrome iron, unlike the iron present in heme group of hemoglobin, is reversibly converted from its ferric Fe3 plus state to its ferrous Fe2 plus form as a normal part of its function as an acceptor and donor of electron.
Another component is risk FES, which exists as a dimer. It is an unusual FES in which one of the iron atoms is linked to two histidine residues rather than two cysteine residues.
Now, as lastly we also coenzyme Q, also known as ubiquinone, is a mobile electron carrier which accepts the electron from complex 1, from complex 2 and from other mitochondrial dehydrogenases such as glycerol 3-phosphate dehydrogenase and acyl coenzyme A dehydrogenase and gets reduced. This reduced ubiquinone is now known as ubiquinol. Electrons are passed from ubiquinol to cytochrome C via complex 3. So what is cytochrome C now? Cytochrome C is a mobile electron carrier which is located in the intermembrane space with the outer face of inner membrane.
So this whole process of transfer of electron via the complex 3 involves cytochrome C1, Bl and BH and a dimer of risks FES and is also known as the Q cycle. The coenzyme Q may exist in three forms: the oxidized quinone, the reduced quinol or the semiquinone. The semiquinone is formed transiently during the cycle.
One turn of the Q cycle results in the oxidation of two reduced coenzyme Q to coenzyme Q, releasing 4 proton into the intermembrane space and the reduction of 1 ubiquinone to ubiquinol, causing 2 proton to be taken up from matrix. So now, are you all ready to proceed with the demonstration of all this transport of electron? Let's go.
Oxidation of coenzyme Q takes place by the transferring of electron to cytochrome B and FES. So there are two fates of electron to be transported through complex 3. First, the electron goes from ubiquinol to 2Fe 2s unit, which further transports it to cytochrome C1. Hence, the reduced cytochrome C1 passes its electron to the mobile electron carrier, that is cytochrome C, accompanied by pumping of 4 protons from mitochondrial matrix to the intermembrane space, developing a potential gradient across the inner mitochondrial membrane. Another fate results in the transfer of electron from ubiquinol to cytochrome B, which further enters the Q cycle.
So this is all about transport of electron across complex 3.
Now group 4 will demonstrate the further transport of electron.
Now The final complex of ETC is complex 4, which is the final stage of electron transport, also known as cytochrome C oxidase, and in this complex, the electrons are transferred from cytochrome C molecule onto oxygen. We have to produce water molecules and also to create proton electrochemical gradient to pass protons in the intermembrane space, which is used by ATP synthase to generate high energy molecules. This conversion by 4 electrons and 4 protons of oxygen to water is unique.
Complex 4 contains 4 redox centers: copper A, cytochrome A3 and copper B. The copper A complex is formed by association of 2 copper atoms. Cytochromes are redox active proteins which contain a heme group with iron as a cofactor. This complex cytochrome A3 is terminal respiratory enzyme of all eukaryotes and many bacteria. Cytochrome A and cytochrome A3 have kinetic distinctions. Functional unit of enzyme is the single protein which is referred to as cytochrome A and A3. The iron of heme A of cytochrome A is hexacoordinated, which means that it is bound by 6 other atoms, while the iron of cytochrome A3 is bound by 5 other atoms, leaving the 6th site available for a peroxide linkage, which we will later talk about. The copper B contains a single copper atom, that too in Cu2 positive state.
The cytochrome C molecule, which accepts electron from complex 3, gets reduced as Fe plus 3 gets converted to Fe plus 2. Then cytochrome C gives its electron to complex 4. Once it attaches to complex 4, it transfers 1 electron to copper A center, and electron goes on to cytochrome A. Then it moves on to cytochrome A3, and that electron that is being transferred converts cytochrome A3 and reduces Fe plus 3 into Fe plus 2. Now what happens that the electron is being transferred to copper B, and then copper B will be transferred from copper plus 2 to copper plus 1. In next step, we have 2 oxygen atoms. This is the same oxygen molecule that we respire in. This is used to form peroxide linkage between heme A3 and copper B.
This high affinity oxygen makes it an ideal electron acceptor. Partly reduced oxygen free radicals are toxic to the cells, and so they aren't released, since oxygen remains tightly bound to the complex 4 between the iron and the copper atoms. Now, because of the other 2 electrons which are carried by cytochrome C, the peroxide bond is broken down. Then, the 2H positive from mitochondrial matrix are extracted, which join to form copper hydroxide group and heme hydroxide group. Then, 2 more H positive are picked from mitochondrial matrix. Which forms 2 molecules of water. So the 2 molecules of water are formed. Now simultaneously, 2H positive ions are also pumped in the intermembranous space. So how these 2H positive ions are pumped in the intermembranous space? So the energy required for pumping H positive comes from transferring electrons, which excites the complex. From this reaction, free energy is harnessed to generate the transmembrane electrochemical potential, which leads to formation of a proton electromotive force, due to which 2H positive pass into the intermembrane space. Now we should also know that the complex 4 can be inhibited by carbon monoxide, cyanide, azide, hydrogen sulfide, which inhibit the cellular respiration. But as these inhibitors are not present right now, this cycle will continue further. Thank you.
afternoon everyone. Today I will be demonstrating the complex 5, the ATP synthase. It is the smallest rotor which is found in the human body. Before starting, I would like certain things which we should know before demonstrating the ATP synthase is that we have divided into F0 and the F1 part. Joining them will be the gamma subunit, which is a subunit of F1 particle. F0 is embedded in the membrane and F1 is found in the matrix. F0 has further subunits A, B, C, and the F1 has further subunits alpha, beta, gamma, delta and epsilon. The F0 mainly comprises of a C ring and a subunit. The C ring has further units which can be from 8 to 14, and the proton enters from the A subunit. The C ring contains aspartate, so the proton will enter the C ring to form an aspartic acid. The aspartic acid is a hydrophobic molecule, so it will start rotating to move towards the lipid bilayer. The further movement of proton will cause the movement of the C ring. This C ring will rotate and the protons will enter the matrix. This rotation of F0 will cause the rotation of gamma subunit. When the gamma subunit rotates, there will be conformational change in the F1. Now the F1 is made up of alpha 3 beta 3 hexamer ring, where the alpha are present to stabilize the beta particles. To demonstrate, we have only shown the beta particles. Beta particles are, we have shown 3 conformations: the loose, the tense and the open state. In loose state, the ADP and inorganic phosphate will be present, and when gamma subunit rotates, the ADP, it will come into a tense state. The ADP and inorganic phosphate will come in close proximity and the ATP will be formed from the open state. So, the B subunit, which was in the F0 particle, B subunit and the delta, they are only there to stabilize the complete ATP synthase. Now certain inhibitors, ephrapeptins are there which bind to the beta subunit and that cause the blockage of the ATP synthase molecule. And oligomycins are there which bind to the F0 particle, which cause the blockage of the proton molecules. Now there is a binding change mechanism which states that the re-entry of protons through F0 subcomplex causes the rotation of gamma subunit, which in turn causes the conformational changes in beta subunit of F1 complex. This was given by Paul D. Boyer, and he also discovered the ATP synthase, the mechanism of ATP synthase. Thank you.
Hello everyone, I am Tarun and I will summarize the process of ETC. Electron transport chain is a series of protein complexes that couple redox reaction, creating an electrochemical gradient to create and release ATP by process called oxidative phosphorylation. It is critical to cellular respiration, produces ATP and also resupplies metabolic pool with NAD+ and FAD+. Absence of ETC causes metabolic stress and dysfunction. ETC is a multi-protein complex comprising complex 1, 2, 3 and 4 with 2 mobile carriers ubiquinone and cytochrome C. Electron moves from higher to lower potential. At the end of electron transport chain, it is transferred to oxygen. This is called biological oxidation. Energy released is trapped as ATP. The NADH and FADH are impermeable to mitochondrial membrane, so malate aspartate shuttle and glycerol 3-phosphate shuttle helps in their transportation across membrane. Some inhibitors of oxidative phosphorylation are atractyloside and oligomycin. Some uncouplers of oxidative phosphorylation are thermogenin and thyroxine. Thank you everyone, and I hope you understood the process of ATP.